All right, A&P folks, here is Chapter 13. We are going to scratch the surface for this chapter. So we are going to focus on a few anatomical components and pieces, and basically for time purposes we're going to skip a lot of what's covered in this chapter and then move on to Chapter 14, which will be the brain. Okay? So our objectives, first off, is to get comfortable with the gross anatomy of the spinal cord, so understand the white versus the gray matter in the spinal cord. Go back into, again, what type of neuroglial cells we'll expect to find in the spinal cord because it's part of the central nervous system. We'll look a little bit about changes as you descend the spinal cord in terms of anatomical features and how the white matter and the gray matter amount and size changes. We'll look at when nerves start coming out of the spinal cord, they form these two roots, the dorsal and ventral roots. So we'll discuss the differences there and what kind of neuron axons are typically coming in and out. And then the protective covering around the spinal cord, which is your three-layered meninges. Okay? We will expect you to also look at a simple reflex, knowing the key components and how they interact and talk to each other to accomplish a reflex, which is something that's accomplished motor sensory interaction without conscious awareness or thought coming into play. So if you look at the organization of the chapter, you would think like, oh, let's start with the brain, but this book actually has us start with the spinal cord and then has us hit the brain. So a lot of what we're going to do here in terms of the meninges and the cerebral spinal fluid is going to tie back into what's also present in the brain. All right, so for anatomy purposes, these are your spinal cord key terms. Your white matter is going to be the part of the spinal cord where we have axons surrounded by oligodendrocytes, which are your myelinating cells, that are going to run the length of the spinal cord. So your axons run the direction of up and down. So think of the white matter as all of these long axons surrounded by their supporting neuroglial cells. They are going to then be arranged normally towards the outer margin of the spinal cord. And so because they are long axons running north-south in direction, when we look at a transverse cut, what we're seeing is all of those axons coming out at us. And if we try to take a gaggle and pull this white matter away, what we're going to find is the white matter is all of these axons running long ways that are columns. All right, so the white matter collectively is the myelinated axons running north and south. If you start to divide the white matter into groupings in the back and the side and the front, you will call it the dorsal or posterior column, the lateral column, the anterior column, and again, it represents all of these myelinated axons that are, in many cases, interneurons that synapse somewhere for descending pathways. Their cell body starts somewhere in the midbrain or upper regions of the spinal cord and end and terminate somewhere usually in the spinal cord in the gray matter, where then the final neuron goes out. And for sensory neurons, the cell body originates somewhere in that gray matter horn area, usually in the dorsal horn, and then its axon runs in the white column areas up, some usually in synapsing, in many cases at the thalamus. Okay? All right. To help get ourselves oriented, fissures are always more pronounced openings in material than sulci. So sulci is going to be these indentations of, if I took a butter knife and put it, this is technically a ditch. It's technically a separation. This material is not connected to this material. They just kind of push into each other. All right, and so there is a physical divot, a depression between these two pieces of tissue. So that's a sulcus. A fissure, on the other hand, is going to be a more pronounced, wider opening between the two pieces of tissue. So for getting ourselves oriented, the posterior is always going to have on midline a posterior or dorsal median sulcus, and our anterior median is always going to have a fissure. So how do I know then what is front and back? I always know that the fissure is on the anterior side, the posterior is the sulcus. And so then at that point I know this is, if I'm standing up, this is my belly, this is my back, So this is, again, my right side. This is my left side. Okay? And you can see the fissure stays pretty continuous through the cervical, thoracic, lumbar, and sacral regions of the spinal cord, and the sulcus is going to be present all through as well. All right, so conus medullaris, filum terminal, and cauda equina. Those are three terms that relate to landmarks of where the spinal cord is located through as a big organ level landmark. All right, so you have the conus medullaris somewhere around this T12L1 region. You have the cauda equina somewhere down here where it's no longer really a true spinal cord. You're beginning to like just have all these white matter axons running outward, and so that's why it's cauda for like a tail, but hair of a horse or something, hair of a horse. That's what it kind of looks like, the horse hair coming out. And then the filum terminal, again, it's kind of a landmark of where the true spinal cord with the central canal with the very organized white gray matter organization kind of ends. Okay. Going back to, again, landmarks in a transverse section, the central canal represents in the gray commissure area where we're on midline, where you have cerebral spinal fluid that's connected all the way back up to your ventricles. Now, dorsal root ganglia, ventral roots. These relate to the spinal nerves that are coming out as part of the peripheral nervous system, right? And the peripheral nervous system is going to have axons that are afferent, so they're coming into the spinal cord, as well as axons that are leaving, that are going out of the spinal cord. All right? And in many cases, this entire nerve is going to be wrapped in epineurium. Then if you look at organization, there'll be some more organization within of some of the neuron and axons wrapped in fascicles, and they'll be wrapped in a connective tissue that is perineurium. And then an individual neuron will have connective tissue around it that'll be endoneurium. And so it's a lot like the muscle where we looked at it. It was epimyosin, perimyosin, and fascicles, and endomyosin. The nerve is very much the same way. And so when we look at some of these fascicles, we're going to gather all these afferent neurons kind of together because they're all going to the same location, and the same with the efferent. And when we look at the afferents that are going to the spinal cord, when we get to this point of the nerve where it needs to separate into the neurons that are going to go to the back door and the ones going to the front door, the afferent neurons tend to head in the dorsal root. So the dorsal root is composed of primarily afferent neurons that are bringing sensory information to the spinal cord. And if you recall, afferent neurons are unipolar neurons. And so their cell bodies are these little leaves that come off of this long axon that then connects to some receptor regions, which are the dendritic arms and the receptor cells that might be the temperature receptors or the stretch receptors, or the pressure receptors, the posinium corposus, the ruffianin, okay? And so all of these cell bodies are going to gather together in the dorsal root, the posterior root area, and because a lot of cell bodies coming together along with all the satellite supporting cells are going to form this term called the dorsal root ganglia. So any gaggle of cell bodies is typically going to be a ganglia supporting with lots of satellite cells. And the advantage of having all these cell bodies here is that it's close to the protection of the vertebra, it's close to the spinal cord, and so these axons, if they get damaged because they're in the periphery, they could regrow with Wallerian degeneration and then, you know, targeted regrowth. And so the idea of, well, at least the cell body has a higher chance if there's damage to the axon to be preserved and keep those sensory afferent neurons alive and functional. The anterior or the ventral root is going to be in many cases where efferent neurons exit. And most of our efferent neurons are going to have their cell bodies in this gray anterior horn region. And the reason why they're going to be there is because in many cases these are your somatic motor neurons that are going out to skeletal muscle and going to synapse at a neuromuscular junction. Okay? Some of the other neurons coming out here are going to be your sympathetic and parasympathetic. And we'll learn more in Chapter 16 that your sympathetic neurons that come out will come in, join the main nerve, and then very quickly go to a secondary gaggle area known as sympathetic chain ganglia. And in that sympathetic chain ganglia, that's where they'll synapse, and then post neuron, or the second neuron in the two-neuron pathway, will then exit and go towards the target organ. Okay, so those are all the key terms that you have to be able to identify in lab, and as well as define in lecture if given a multiple choice question. Okay. Now, the spinal cord does not physically touch bone. So there is a protective covering that separates the periosteum of bone and the spinal cord material of neurons and neuroglial cells. And that special tissue is known as your meninges. Your meningeal layers are 3 layers. The outermost layer, which will connect into and be closest to the periosteum of your spinal vertebra and your skull bones, is going to be your dura mata. Now, in the spine, the dura mata and the periosteum have a little bit of space, and so that space is known as epi because it's outside, dura, the epidural space. And so when you hear about people getting an epidural, that's where the doctor is trying to get the needle into. It's trying to get it into the space that's between the periosteum and the dura mata. And in the spinal cord, it's a more pronounced space, and there's usually some adipocyte cells that live in that region. All right? In the brain, the epidural space in some regions is non-existent because the dura and the periosteum are going to be more merging into each other. And so the dura mata, because it's the outermost layer, is going to be your most collagen, elastin, toughest layer of the 3. All right, the next layer is going to be the arachnoid mata. All right, and the arachnoid mata is going to be tied into your cerebral spinal fluid. Now, between the dura and the arachnoid mata, there is a little bit of space known as the subdural space because it's underneath the dura. And so if you go back and you read about Bret Michaels and how he got his head hit in one of those Tony Award presentation performances, and then he ended up with a subdural hematoma, he had a bleed and a collection of blood in the space between the dura mata and the arachnoid mata, which is the subdural space. And so you sometimes will hear about subdural hematomas, which is a bleed and a collection of fluid that ends up in this little space between these 2 membranes. And it's a very small space, okay? So if your epidural gets put in a little too much, it'll end up releasing drug not in the epidural space, but potentially in the subdural space. All right? Now, the arachnoid mata has a space between it and the pia mata, which is known as the subarachnoid space. And that area is not going to be like the other spaces we've talked about because it's going to be where cerebral spinal fluid fluctuates. So this cerebral spinal fluid is connected back into the cerebral spinal fluid that's in the central canal and the ventricles, and it has a little access point near the inferior area of the 4th ventricle before it connects into the central canal that allows some of that fluid to actually go into the subarachnoid space. So when you hear about people having their spinal fluid tapped, this is the place the needle's trying to get to. It's trying to get to this little area so it can pull out cerebral spinal fluid and not blood from the dura area or the subepidural area. Okay? And the pia mata is the innermost, and it's going to be the part of the meningeal layers that's physically kind of like a periosteum, a perichondrium, that's connecting, touching, interacting directly on the outer region of the nervous tissue. Okay? So when you read your book, again, you'll go into more details of the dura mata is the outermost layer. So it has, so if you look at the brain, there's skin, there's a periosteum, then the bone, then another periosteum, and then you get to the dura mata. And the dura mata will, in some areas, not have much of a epidural space. In the spinal cord, you'll see that the dura mata does have more of a epidural space with that fat cell population. And again, that first layer of the meninges needs to be your strongest, most protective layer. So it's going to have some dense collagen fibers, and it's going to have, again, some strength to it because it physically is starting to be the first protective layer around underlying neural tissue. Between the dura and the arachnoid will be a subdura space, and again, that subdura space will be pretty small in most cases, all right? And then you get to the arachnoid mata. And the arachnoid mata is going to, again, be some simple squamous epithelium, somewhat like a mesothelium of a visceral parietal peritoneum pericardial membrane. And that's going to be part of how it lines and keeps in the subarachnoid space cerebral spinal fluid. All right? Now, this is not just like a fluid-filled space. There is some trabecular type material, so there are some collagen and elastin proteins that push in and connect the arachnoid mata and the pia mata to each other and provide a little bit of structure to that space. All right, and so you'll see over here, again, it's still pretty thin, and then you'll have some subarachnoid space, so right in this region, and there's still that trabecular material that kind of gives the cavity a little bit of like tent poles to hold it up. The pia mata is going to be a meshwork of elastin and collagen fibers directly touching or the closest to the cortex, the gray matter of the cerebrum, of the brain material, and the white matter of the spinal cord. All right? And you'll see that, again, blood vessels can run in certain regions of the meninges, but the blood vessels don't technically run into because of that blood-brain barrier, and part of that comes with the meninges helping, and then the neuroglial cells providing a little protection of those arachnoid cells and epidermal cells. So the blood vessels don't actually go and become part of the gray or white matter of the nervous tissue. Okay? Now, white matter is going to usually be areas of neurons that are myelinated and high with fat. So myelin is a fat molecule that gets made and high density in your neuroglial cells that then wrap around your neuron axons that are propagating electrical impulses. And those electrical impulses, remember, are just small changes in ion concentrations across the cell membrane. So in the spinal cord, the white matter is going to be your external part of the organ. So your material running north-south, running and connecting until the brain to potentially the final output or initial input neuron are going to be located on the margins of the spinal cord. Where the gray matter is located, that's where you have axonal cell bodies, dendritic surfaces, and terminating axons. All right, so this region is where more of synapses between neurons are occurring. And what we're going to find is the butterfly kind of feature in the spinal cord gives a pointy part towards the back, a pointy part towards the front, in some regions a pointy part that kicks towards the lateral surfaces. And so the points of the gray matter that tend to be where cell bodies are located for efferent neurons, synapses are located for afferent neurons, and cell bodies for interneurons are going to be your posterior horn, your anterior horn, and your lateral horn. Okay? And there's less myelination here because we're not looking at the axons and large portions of the axons, just small portions of the axons, and most of it being this other parts of the neuron that are not necessarily myelinated for function. Now what's cool about the spinal cord is everything is very, very organized. And it's so organized that in research we can potentially damage a small, let's say, quarter of the so if we were to inject acid and damage just this quarter portion of the spinal cord, we would then expect to see sensory problems at level. So if this is at around T3, so we would expect to see on that damaged T3 region of the spinal cord sensory issues occur because we damaged sensory input areas, which is the posterior part of the spinal cord. If I go and I potentially damage, with an injection, the front quarter of the spinal cord, I know then for that T3 level, I'm potentially taking out all of the motor ability that comes out of the spinal cord at that level. So I might be paralyzing certain parts of the body for whatever at T3, and then because I'm taking the white matter out, whatever columns I'm affecting below level, I might be paralyzing from that point down all of those muscles. So that's the cool part of the spinal cord, is everything is super, super organized. All right? Most of what is in the back is sensory. So any lesion, any damage is usually going to be sensory related if it happens to the posterior back side of the spinal cord. If it's on the right side, there will typically be damage to some right side level sensory, and then for below level and on the left side, some below level left side sensory. So sensory crosses over pretty quickly in the spinal cord. For motor, the anterior part is where most of somatic motor and autonomics come out. And so any damage I get in a spinal cord, a spinal lesion, a spinal bleed, a spinal tear that's in the front or the anterior part of the spinal cord is going to translate to potentially at level and below motor problems, okay? And then we can even go one step further and even look at the gray matter is divided in. Most of the synapses on the very tippy part of the posterior horn are sensory nuclei from first the around the muscle, around the skeletal muscle, so stretch reflexes, Golgi apparatuses that kind of protect agonist, antagonist, so you don't pull your ligaments and tendons off the bone. And then most of the sensory inputs from the abdominal and the inner regions and around the visceral organs are located more into the inner segment of the horn. For the motor, most of the anterior nuclei out in the anterior horn is where motor neurons connecting to skeletal muscle are located. And then the autonomic cell bodies for parasympathetic, sympathetic neurons leaving to go control smooth muscle are going to tend to be on more of that lateral horn part. So it's really kind of cool how that's organized. When you look at the white matter, there's an organization as well. So typically speaking, the things that are going to be located in the columns, like the arm might be located here, and then it's like the wrist, and then the thumb. So there's an organization even to like which columns, which axons are bringing sensory information from certain areas of the body, and then it feeds into, when it gets to the brain, that it goes to specifically areas of the brain that are also body mapped. So the body mapping of the brain also is body mapped on the spinal cord. So again, if you really get into neuroanatomy, if you only damage just this region of the white matter on the back of the left spinal cord, you would potentially then be able to pinpoint exactly where you're potentially taking away pressure, temperature, pain, sensory inputs. And so you would potentially have an area of your body that you could pinpoint and poke and prod, and your body would never feel it because that part of the spinal cord is damaged that carries that information. All right, so the last thing we need to talk about is the spinal reflex. So there are lots of different spinal reflexes, and all of the spinal reflexes exist because there are some times where we need a stimulus and a reaction to that stimulus to occur before conscious awareness or the brain gets the information of this is happening. So if you're cutting yourself or burning yourself or doing damage to yourself, you want to potentially have a muscle response to that before you get awareness in your brain that you're in pain or you have a problem. And so most of the time your spinal reflexes consist of these 5 components. They have some type of receptor. So it could be a pain receptor, a temperature receptor, a touch receptor, a pressure receptor, a stretch receptor in the muscle. And that receptor gets hit by a stimulus. And the stimulus is pretty strong, and so on the sensory neuron, the afferent unipolar neuron carries that information to the spinal cord. And in many cases, it's going to synapse. Now, remember that it can potentially synapse with 2 or 3 axon terminals. And so one of the synapses is going to be on an interneuron that is going to take the information from the posterior horn to the anterior horn. And remember, the posterior horn is where we have somatic sensory neurons coming in, and the anterior horn is where we have somatic motor neurons that exit and go out. So very quickly you can get the sensory information relayed to motor pathways that allow contraction on a motor cell to occur that will lead to, if I'm burning my hand, my withdrawal reflex. If I'm contracting too much of my bicep muscle, then I could maybe counteract and contract a little bit more of my tricep muscle so I don't rip my elbow joint, you know, when I'm moving it. Okay? Now, technically speaking, there's another interneuron that'll exist, and this other interneuron is eventually going to take as a synapse and probably cross over and carry sensory information up in those dorsal columns. And that information is heading to the brain. It's just that by the time the brain gets in the post-central gyrus, which is the primary sensory cortex region for that maybe area of the body, gets awareness that I'm burning, I'm in pain, I'm cut, the muscles have already contracted and pulled you away or tried to remove you from that stimulus, and your brain is not even consciously aware that that's occurred yet. But when you get awareness a few microseconds later, you'll be even maybe more of, oh, I need to back up, or oh, I need to get away from the stimulus. So the reflex is these components. Realize that there is going to be information that heads to the brain on a different interneuron, but it's going to reach the brain and come into conscious awareness after your body's already taken at least some small, non-conscious, non-aware reaction to that stimulus. Now, most of your reflexes have the integration interneuron except one. The most simple reflex is the knee-jerk reflex. So that's when you like bang on your patella ligament and then the foot kicks up. That's the simple reflex in that the sensory neuron actually can directly talk to the effector neuron and get the contraction of the quadricep muscles to extend the knee before you flex and tear the patella tendon and the patella ligament. Okay? So that's the only knee flex reflex is the only reflex that doesn't have number 3. It has everything else but number 3. All of these other reflexes have the 5 components. Okay? So as we look at the spinal cord, again, the spinal cord has a narrow central canal that's connected to the ventricles and full of cerebral spinal fluid, and then it has a subarachnoid space that's full of cerebral spinal fluid. So like the brain, it's completely embedded in fluid, and that helps when you jump around and do different things that your spinal cord doesn't slosh around and get bumped and damaged against the bone that it's encapsulated in. The areas between the bone are going to be where the nerves come out, and typically the neurons are going to come out of the front and into the back. And so it's going to form a dorsal and a ventral root, and those roots will then combine into a spinal or cranial nerve. Normal organization of the spinal cord is sensory comes in the back, efferent, motor comes out the front. And then you can go even further in the white columns that are in the dorsal region tend to be sensory pathways that are ascending to the brain and eventually to the sensory cortex region to bring information on sensory from the body. Your anterior columns and some of your lateral columns are the areas where your descending motor pathways are typically held, and those are the interneurons that eventually synapse on the anterior horn, and the motor neuron that then leaves and goes and has at the neuromuscular junction an effect on skeletal muscle. All right, gray matter, it tends to be the area that is in the middle of the spinal cord covered with white matter. It's non-myelinated, it's cell bodies, it's dendritic surfaces, it's interneurons, but there's lack of large amounts of oligodendrocytes and large myelinating areas. So that's why it gets more grayish in color when you stain it versus the white matter, which is very lipid-rich in myelin with those high-density oligodendrocytes with their arms wrapped around the axons. So that's why it stains white. Again, there's a lot of organization, and so it's really cool if you want to get into the neurology, the neuro pathways, the neuro ascending, descending columns. We can pinpoint if we know the damage is at T4 nerve on the anterior front right quadrant, that is going to mean this damage is from that level and below going to be seen. So if it's the complete right side of the spinal cord gets damaged, we know we're going to have sensory and motor issues because sensory's in the back, motor's in the front. If it's only the dorsal or posterior part of the spinal cord, we know we have all sensory issues. If it's the front, we know we have all motor issues. So it's really, really interesting. And there is some really cool research out there where they do just that to rats and mice. They inject with a needle into the spinal cord, release acid, kill a part of the spinal cord, and then they know that the reaction is going to be either motor or sensory depending upon where the needle was releasing that acid. Again, reflexes are defined as these automated responses where a stimulus is needing to have a reaction occur before the brain gets awareness, and so those reactions are built in to usually protect because the stimulus is such that it might be causing harmful damage. So that's class, you guys. That's chapter 13. We'll get to chapter 14 soon enough and hit the brain. I'll see you next time.